So hello, everyone. It's my pleasure to introduce to you Feyza Özgen. Feyza was born and raised in Istanbul, Turkey. And at Istanbul University, she studied genetics and bioengineering. And after that, she was working for 2 years as a research assistant in the field of protein engineering. And in 2018, finally, she decided to start her PhD. And she was moving from Istanbul to Graz in Austria, where we started to work together within the innovative training network Photobiocat. And Feyza's main part of research back then basically focused on looking at possibilities to drive risk-type hydroxylations by light. And last year, after it became obvious that I will move to University of Groningen, luckily Feyza decided to move with me, which I much appreciate because Feyza is doing a really great job and she's a great personality to have in the lab and to work with. And yeah, since last year, we continue our research on light-driven light-driven reactions here in Groningen. And today, Feyza will talk a little bit about a collaborative project that we have initiated with a partner from the University of Pavia in Italy, in where we are looking at a photocatalyst that is catalyzing a CC bond formation reaction with an enzymatic reaction step to produce chiral delta lactones. So, but I don't want to anticipate too much. And with that, I will give the word to Feyza. And Feyza, we are looking forward to your talk.
As you might understand from the cover slide, I will talk about light photobiocatalytic systems. Working with light is quite cool, but it's sometimes very challenging. But we accept this challenge and to harnessing the power of light to drive photobiocatalytic reactions. So in my project, the concept is PHOTO-CHEMO-ENZYMATIC cascades reactions with the implementation of light, of course. In those cascades, photocatalysis is combined with the enzyme catalysis. If you think about that, actually you are kind of combine two different worlds. But this cascade mechanism is actually can be really strong and can enable powerful reactions. If you look at that, you start with the substrate to convert to the corresponding ketone or aldehydes, and which should be functionalized to desired product. For this such a cascade reaction, first you need light and you need organophotocatalyst for the first step of the reaction. And then, of course, you need enzyme to functionalize the ketone to, for example, amines or nitriles. So I would like to show you some examples of photo-chemoenzymatic cascades. Here you see the sodium anthraquinone sulfonate SAS. It's a photoorganic catalyst. It's very popular catalyst for this such a cascade reactions. In the first example, Frank Holman and co-workers developed a one-pot sequential cascade combining light-driven oxyfunctionalization reaction using SAS and subsequently highly selective omega transaminases for biocatalytic reductive amination. However, at the beginning, they face a problem in a one-pot or one-step procedure, like inactivation and degradation of biocatalysts by photoexcited SAS. And therefore, they establish one-pot, two-step reaction mechanism, and with that, they reach high yield and high EE value. In the second example, they again use SAS as a photocatalyst, and then they perform the enzymatic reaction using HNL and BAL enzymes. And here they run the reaction in semi-preparative scale and in a sequential running mode, with yielding nice isolation of the product. And as I said, they perform in sequential running mode using 2 steps, 2 phases in order to have less introduction between photocatalyst and enzyme at the end. Now I would like to show you another photocatalyst called TBADT. It's a tungsten photocatalyst. It's very challenging and unique. It can perform CH-CC bond formation that also allows the generation of radical under very mild conditions. And for the reaction, you can just use small amount of this TBADT. You see the very complex structure, and it can also remain active for many catalytic cycles. Here you see the TBADT reaction before and after, and for the oxidation of the photocatalyst, you need near UV light. And this characteristic blue color actually is very cool that you can track your reaction, that you can monitor is it working or not. Here on the right side, I would like to show you an example, the mechanism of TBADT. Actually, it's performed by our collaborator that we are together in this project. So basically, sequence starting with aldehyde with electropoor olefins in the presence of TBADT. So upon light irradiation, the CH bond of heptanaldehyde was cleaved by TBADT, which is in an excited state to give an acyl radical. And then this reacts with the ethyl malates to radical adducts, and then hydroacylated the derivative. And during this process, also the reduced form of photocatalyst can be regenerated. So it's very cool photocatalyst. So I'm just showing you the general concept. And so because of the nice feature of the TBADT, we got interested to catalyze the CC bond formation, and then the reductive reduction with the alkyl dehydrogenases to the desired products. And if I would like to show you the goal with more detail. So now you are seeing the exact substrates for the first step of the cascade reaction. I use a heptanal triphenyl propanol with the combined with methyl acrylate under UV light irradiation. And I hope that to obtain the ketones. And after obtaining the ketones, of course, our enzyme, the alkyl dehydrogenases, will take role to produce main product, which is actually spontaneous generation of lactones. For this cascade, photo-chemo-enzymatic cascade reaction, of course, first we wanted to screen some alkyl dehydrogenases. So I tested 4 different alkyl dehydrogenases from different organisms, and also did the activity test with the ketone that already sent by our collaborator. And you can see on the graph that we were really happy that we identified active enzymes, and actually at least 2 of them show quite a high activity towards these ketones. And of course, we did extra stability test of these alkyl dehydrogenases in the presence of aldehydes and simple acrylates, because at the beginning the idea is to perform this cascade simultaneously. And after all the results, we decided to do initial study, just testing with the alkyl dehydrogenase from Rastonia. So for the cascade reaction, of course, firstly, I need to synthesize the ketones. For that, how I'm performing the reaction. So I need of course, photocatalyst needs to dissolve in the reaction media. Here, in my cascade system, I use the reaction media having a 35% acetonitrile. Normally, in literature, they were performing this reaction in the presence of 100% acetonitrile. But of course, I needed to think about the enzymatic step. So I decreased, and then adding the aldehyde and methyl alkylate, and of course using the UV LED strips at 365 nanometer. And after the 20 hours later, we were able to form the corresponding ketones. Here you see this is the aliphatic ketone and this is the aromatic ketone. And the product concentration, yeah, we saw that the reaction is working, but the product concentration was not so high as we were estimating, because we didn't use the high percentage of acetonitrile. And additionally, I wanted to also to test the different concentration of methyl acrylate, and when I increase the acrylate concentration, then I reach a more product concentration of ketones, which may be because of the volatility of methyl acrylate, and also the reaction model was different than the literature. And then we assume that the ketone form in our pot. And then our enzymes take roles, and we edit alkyl dehydrogenase Ras ADH into the same solution, and of course switch off the light. And then later on, we were able to form the corresponding ketones. Here is the outcome. This side showing the EE and this part showing the total lactone concentration. If you look at that, if I increase the percentage of organic solvent, of course, I reach higher lactone concentration. And when it's low, it was, of course, low. And additionally, we saw that the EE was low in the case of Ras ADH. And also, we observed that there are still alcohol remaining in the solution, so the lactonization was not over yet. So we got this data at the beginning, and we decided to optimize the system more, and also test go back to other alcohol dehydrogenases can show the high enantiomer selectivity, and also we wanted to work on the lactonization to shift it to 100%. And for that, first we started to test the other different alcohol dehydrogenases. And I chose for oxidiconic acids with methyl ring butyl substitution and with phenyl ring, sorry. And then implement the cofactor regeneration into system to yield chiral gamma decalactones. If we look at the first one with the ketone with methyl group, that we obtain really nice conversion. And of course, for Ras, we knew already that the selectivity was low. But in case of SMCR M4 mutant, then we reach really high enantiomer, which also knew it from the literature, because this ketone already studied with the SMCR mutant. In this second part, when we tested with the ketone with butyl group, the conversion in this case was low. The orality was low, but for the SY-ADH, then we reach really good selectivity. In the third one, with the phenyl ring, the conversion again was high. And for SY-ADH and for LK-ADH, we reach really good selectivity and enantiomer selectivity. Then, as I said in previous slides, that I need to do also to shift the lactonization to 100%. Therefore, after performing the reaction, then I stop it by adding sulfuric acid and running extra 1 hour at room temperature. As you might see on the graph, that the alcohol is just turning to the lactone, and then we were able to complete the cyclization. After these results, then we were ready for the cascade reaction. But this time, we also optimized the first step. What we did in the previous slide, I show you that we were using 35% acetonitrile for the enzymatic step that thought that can tolerate. And but here we wanted to also have a higher product concentration. Therefore, we followed exactly the procedure of according to the literature, using also testing 80% acetone, which shows the blue bars, and the 100% of acetonitrile. And that's for that reaction, I use heptanol, testing methylacrylate, butylacrylate, and phenylacrylate. And as you see, we got really nice product concentration. Then we decided to dilute this TBADT reaction for the enzymatic step. So now you are seeing the whole PHOTO-CHEMO-ENZYMATIC cascade and testing with the 4 different alkyl dehydrogenases. As I said, I decrease the TBADT mixture in our main cascade reaction to 6%, and I perform the reaction in 80% acetone and 100% acetonitrile that I already prepared. And after the cofactor generation system and using cell-free extract containing alkyl dehydrogenases, and additionally also the acid addition, we formed chiral gamma lactones, especially for the SMCR mutant. We reach 95% conversion and 96 LR enantiomer, and in the presence of 80% acetone. And when we tested the media prepared in 100% acetonitrile, the conversion was really, really low. Actually, we also knew it from literature that they already tested the effect of different organic solvents, that acetone is much, much better than the acetonitrile. And for the other alkyl dehydrogenases, the acetonitrile was better than the acetone mixture. And for the second one, with the reaction with prepared with butylacrylate, and then we again saw nice results. But as we already observed in the enzymatic reaction, the conversion was low. And for the SY and LK, we reach and also good enantiomer selectivity. In the third one, that with the phenyl ring, we perform again reaction in acetone and acetonitrile. And for Rus alkyl dehydrogenases, this time we reach really full conversion. And for the SY and LK, the conversion was low in acetone, but a nice AE value. But when we switch to the acetonitrile, then we reach really high conversion and also perfect enantiomer selectivity. So those are the results that we got so far, and we are actually quite satisfied about these results because we reach really nice yielding chiral gamma decalactones and high enantiomer purity for some cases, but still we need to look for optimization to make the system a bit more better, to improve the system, maybe testing different wavelengths and also increasing the enzyme concentration can help for the optimization a bit more. So with that, I'm coming to the end of my presentation. Of course, I would like to thank firstly to my supervisor, Sandy Schmidt. It's a great pleasure to working with you. I'm really happy to having a chance working in such a cool project with you. I would like to thank to our collaborators, David Ravelli and Luca Capaldo, contacting us to giving an idea of the TBADT is such a cool cascade mechanism. I would like to thank to other supervisor, Gerrit and Robert, the technicians, Peter, Ronald, and I would like to thank to my lab colleagues, especially to Michael Ronda. He is a really great lab partner, and I would like to thank other lab members, the ITM, the Marie Curie, the ITM project, the Photobiocatalytic project that I'm in. And lastly, I would like to thank you to you all for your attention. Happy to hear your questions.
Great. So, go ahead, Andrew. I was just going to say thanks so much for a great first talk, Feyza. And Andrew, do you want to go ahead and lead off the Q&A?
Yeah, so it looks like Anya actually has a question, so I'll let her ask first if she wants to turn on her microphone, and then we can move to the Q&A box.
Hi, Feyza. Thanks for a great talk. I was curious whether you consider taking this cascade even further and replace the cofactor regeneration by a photocatalytic cofactor regeneration that a lot of groups has been working on.
I mean, yeah, I tried other also the systems, but at the end we found that the best ones was the glucose dehydrogenase from Bacillus merotherium, because we were also thinking about the stability of the enzyme. So yeah, I think we will continue with this.
Yeah, thank you.
So a quick question from an anonymous attendee asking how the TBADT is generated. How's the catalyst made?
I'm not quite familiar with the mechanism. I wish I had the system, but it's a general radical chemistry. It's the hydrogen atom transfer mechanism. So, but I will look into detail, of course.
So this question came up at a previous meeting too, I guess, defining what we call an enzymatic cascade. The question asks, they wonder if the scheme can be called a cascade if the reactions are separated in fact. So I guess this is more a nomenclature question, but what are your opinions on that?
Yeah.
Okay.
Maybe I can also comment on this, Andrew. Is that
sure.
Okay?
Yeah.
So, of course, in a way, it's not really considered as a true cascade because it's not, or we are not yet able to perform this simultaneously. But we're still trying to work on a system that can at least run in one pot. It's then still sequential, but at least in one pot and not in two pots. So yeah, I fully understand that question and that concern also.
Yeah. We have another question here. They were wondering, what would your guess be as the rate limiting step in your cascade reaction?
Sorry, I didn't get the question.
Oh, sorry. They said, great work. Wondering what your guess would be as to the rate limiting step of your cascade reaction.
What step? I'm sorry, I missed.
Oh, the rates limiting step? Like
Okay.
what?
The problem is, as I mentioned, this is a different roles that we need to find a good solvent for both catalysts and yeah, I mean, it's very quite challenging, and there are some limitations, but for the stability, maybe we can work with some immobilization studies and maybe biphasic systems that can that we can maybe overcome some limitation problem.
And we have one last question. They want to know if the catalyst can activate non-aldehyde compounds to add double bonds. Can you use this catalyst for other things?
Oh, okay. I'm not sure about this. I think I need to further experiments to figure it out.
So maybe I could also add something to this. So yeah, that's possible. You can also use this for sp3 hybridized carbons, but it's less efficient. So with the aldehyde functionality, it goes actually really quick, and it's really fast, the reaction.